Hi students, we had previously spoken about mutations and now we are going to talk about mutations in details, especially the induced mutations. So let's go into it. Induced mutations are those mutations, of course, which are being caused by certain added substances. That is, they do not naturally occur, but they are being caused by the presence of certain external agents in the DNA. Usually, mutagens are the name of the substance which causes a mutation. And any agent that damages the DNA, alters its chemistry, or in some way interferes with its function will probably induce a mutation. There are different types of mutagens, basically the chemical mutagens and the physical mutagens. Chemical mutagens, they include base analogues, alkylating agents, deaminating agents, hydroxylating agents, as well as the intercalating agents. Among these, the alkylating agents, hydroxylating agents, and the alkylating agents can commonly be called as the DNA modifying agents. And among the physical agents, you have basically two types of the ionizing radiations as well as the non-ionizing radiations. And among them, the ionizing radiations include the X-rays, gamma rays, etc. Whereas the non-ionizing radiations mainly include the UV rays. Now let's start with the base analogues. The base analogues, these are chemical molecules, okay, and they are structurally similar to the normal nitrogenous bases, and they can be incorporated into the growing polynucleotide chain during the replication. As the term implies itself, it is an analog of a base. That is, it is similar to that of a base. And once in place, these compounds typically exhibit the base pairing properties different from the bases they replace and can eventually cause a stable mutation. Now, in the case of 5-bromouracil, it is an analog of thymine whose enol tautomer forms hydrogen bonds like cytosine, pairing with guanine rather than adenine. Okay, so the analogs have a problem is that though they are analogs, their base of bases, what happens, their base pairing rules will be changed. In the case of 5-bromouracil, okay, 5-bromouracil, it is an analog of thymine, okay, and in the keto form, it will form base pair, it will base pair with adenine, but in the enol form, it will base pair with guanine rather than adenine. So, as we saw earlier in the previous sessions, when the wrong base pairing happens, that would result into the mismatching as well as mutations in the DNA. Coming to the DNA modifying agents. The DNA modifying agents, as we mentioned earlier, they are of different types. You have alkylating agents, you have deaminating agents, you have hydroxylating agents. There are different types of DNA modifying agents. So the mutagens that change a base structure and therefore alter its base pairing specificity include the DNA modifying agents. And these mutagens, some of them are selective and they preferentially react with certain bases and produce a certain kind of DNA damage. Now, in the case of methyl nitrosoguanidine, it is a alkylating agent and it adds methyl groups to guanine, causing it to mispair with thymine. So, depending upon what type of modifying agent it is, the modification that these agents do to the DNA will vary. In the case of an alkylating agent, the methyl group is being added to the base and mispairing will happen. In the case of a hydroxylating agent, a OH group will be added and a deaminating agent will remove the amino group from the base. And ultimately, in all these DNA modifying agents, they will cause the mispairing with their nitrogenous bases, corresponding nitrogenous bases. Now coming to the intercalating agent. Intercalating agent is something which will go in between the DNA strand. Okay, they mainly distort the DNA to induce single nucleotide pair insertions and deletions. And they are planar and insert themselves or intercalate between the stacked bases of the helix. And this could result to a mutation, possibly through the formation of a loop in the DNA. Now, there are different types of intercalating agents. You have now proflavin as well as acridin. These are intercalating agents. if this is a DNA strand that you have, a double-stranded DNA, okay, uh, this is one strand and this is one strand. You have the bases here, which they are pairing. Base pairing is happening. What the intercalating agents do is they will squeeze into the two strands of DNA, okay, and that could result in the formation of either deletions as well as duplications as well as additions. So when the dye intercalates between the double helix, what happens? Mutations in the form of deletions, duplications as well as additions. Another agent that we have to talk about here is ethidium bromide, which is of much importance in our molecular biology labs. Okay. And ethidium bromide is mainly used as a DNA visualizing agent after doing the agarose gel electrophoresis. Ethidium bromide intercalates between the DNA, and hence when we need to analyze the presence of DNA on agarose gel, what does it mean? We mix the samples with ethidium bromide, and wherever the DNA strand goes and moves and forms where it locates on the agarose gel, this ethidium bromide will intercalate. And when you expose the agarose gel against UV light, what happens? The regions where the DNA strands have been present will show a fluorescence. The fluorescence is mainly due to the is a property of ethidium bromide. Since ethidium bromide intercalates between the DNA, what happens? Wherever DNA is present, there ethidium bromide would also be present and they would fluorescence when they are being exposed to UV light. But we should remember that ethidium bromide is a carcinogen and it causes also mutations within us. So thus, what happens? You have to handle them with much care. And you should take the precautions when you are visualizing a gel after staining with ethidium bromide. Now coming to the physical agents. There are basically two types of physical agents. One is the ionizing radiations and the non-ionizing radiations. The ionizing radiations mainly includes X-rays, gamma rays and the alpha rays, whereas the non-ionizing radiations mainly include the UV rays. The UV rays, they are mainly responsible for the formation of thymine dimers. Okay, the dimers are being generated when the nucleotide bases are being exposed to the UV rays. And the same principle is being used in the case of when we in the laminar airflow, you have a UV light and we expect to kill the microbes by using the UV rays. The process of killing of the microbes using UV rays is being done, expecting for a thymine dimer formation, and that would be lethal to the microbe. Now, other than the radiations, the UV radiations, we also have certain other physical agents which could cause, and also certain other carcinogens which could result in the formation of mutations. Now coming to the ionizing radiations and DNA damage. The ionizing radiations, of course, it mainly includes the X-rays, gamma rays and all that. And the extent of their damage varies upon the dose at which we are exposed to them. Now they mainly produce free radicals that not only damages the DNA but also the proteins and the lipids in a cell, and the rapidly dividing cells are prime target for the ionizing radiations. However, the severity of the damage depends on the dose of the radiations. I think it is quite evident for every one of you all. When you go to a hospital to take an X-ray, they say that pregnant ladies be away from the X-ray room. That's mainly because the X-rays are quite damaging, and even the fetal damage could happen when an X-ray is being exposed to the fetus. So that's the reason that we say that you should be away from an X-ray room. Okay. Now these X-rays can cause cross-linking of the DNA or the protein, and it can cause chromosomal breakages, strand breakages, chromosome loss, and also deletion of the bases and the DNA strand breakages. So now you can look at this picture to get a better idea of the severity. You can find that, see what happens, the hydrogen bond breakage can happen. Even the double strand breakages, that is, this is a strand of DNA. and this is a strand of DNA. So the double strand DNA breakages can be possible by ionizing radiations. Sometimes what happens, pyrimidine dimers might be formed. Okay, the base loss can happen, then base change can happen, and DNA cross linkages can happen. Then cross linkages between bases can happen, then single strand breaks can happen. So, and all the severity of the ionizing radiation depends upon the what? The dosage of the ionizing radiation. At a molecular level, the lethal dose of X-ray, for example, 350 to 500 REMS, breaks the phosphodiester bonds between the DNA and results in the strand breakages. So, it depends upon the extent to which you get exposed to the ionizing radiations. And some terminology which I would like to say is about teratogens. These are mutagens which could cause the congenital malformations. Carcinogens are the mutagens which could cause cancer. And the clastogens are the class of mutagens which are responsible for chromosomal breakages. So I hope the portion is clear to you. Thank you.